Welcome back, everyone. I hope you are doing well. Today, we welcome Dr. Samuel Reiter as our speaker. Dr. Reiter completed his PhD in neuroscience through the NIH Brown University Graduate Partnership Program at Brown University. He then completed a postdoctoral fellowship at the Max Planck for Brain Research in Germany before joining the Okinawa Institute of Science and Technology in 2019. He is currently an associate professor and head of the computational neuroethology unit at YST. His research combines experimental and computational approaches to understand the neural basis of animal behavior. Using cephalopods, including octopus, cuttlefish, and squid, as model systems, his work investigates visual perception, camouflage, sleep, motor control, and collective behavior, with the goal of uncovering general principles of how brains generate behavior. Today, he will give a fascinating talk about cephalopods as biological image generators. Thank you again, Dr. Reiter, for joining us, and you can start on your own. Thank you.
Thank you very much. Thank you for giving me the opportunity to tell you a little bit about our work, and especially, coming from someone who's living on a small Pacific island, it's great to be able to do this online. So today I'll tell you about cephalopods as biological image generators. So take a look at this image of the Okinawan reef. If you're good, you can see the octopus in the image. Okay, so on the right here, I've taken 4 crops from the image on the left, and I want you to try to identify exactly where each one of these crops are from the crops on the right are in the image on the left. Okay, I think you're going to find that that's a difficult problem. If instead I ask you roughly where are these 2 crops from compared to these 2 crops, I think this is trivial. This is, that these 2 crops come from the foreground and these 2 crops come from the background. So I think the difference in difficulty level between these 2 tasks tells us something interesting about our visual perception. It tells us that it doesn't work by comparing images pixel by pixel, but instead works by extracting a set of summary statistics that comprise different regions of the visual scene. And this statistical summary is known they're known as visual textures. Now, decades of work have shown that visual texture perception is really important for a quick understanding of a scene, and it sets up later more computationally costly operations like object detection. Now, it seems that visual texture perception is not only present in humans, but also, I think, explains why animal camouflage exists. So take a look at these 3 toads. They're leaf litter toads from Panama. They don't look like leaves in terms of pixel by pixel exactly matching a leaf. Instead, the camouflage pattern resembles the statistical properties of leaf litter. And the predator of these animals must be using visual texture perception to analyze the scene. If instead they were using a pixel by pixel detection mechanism, then they would immediately identify these toads. The toads would get eaten, and the camouflage would never evolve. So I think that the fact that we see camouflage across the animal kingdom tells us that the visual texture perception is also present very widely across animals. Now, the best camouflage by far in animals is found in the cephalopods. These are the coleoid cephalopods, octopus, cuttlefish, and squid. And they're able to change their camouflage in so-called active camouflage, so change their skin pattern to match really precisely different environments underwater. Now, they can do this using their brains to control their skin patterns, and we'll describe how this works in a little while. But this behavior is really fascinating for a number of reasons, and let me explain a few of them. So I think the most interesting thing is that this behavior, these animals are giving us a report of their visual perception in terms of an image displayed on their back, ready for us to observe with a camera. So they don't need to they don't need to be trained to do this. They're just doing this all the time, especially in the wild, but also in the laboratory. Um, and they can really fool our visual systems. I mean, this cuttlefish in the middle of this image, it's really precisely matched to the coral reef. Now, uh, this behavior has evolved to fool the visual systems of their predators, and their predators are diverse. They're fish and marine mammals and birds. The behavior also has evolved to fool their prey, so to disguise them from their prey, so which are include many crustaceans. Likely, this behavior has evolved to fool diverse visual systems, and it even works on our own visual system, even though we're not the historical predator of these animals. So, if we can understand how to adopt in which situations, I think we can learn something very deep about visual perception in general, not just about the visual perception of these amazing animals in particular. Also, this behavior seems to be somewhat simple. These animals can do camouflage from birth. So here's a 6-week-old cuttlefish, excellently camouflaged to tiny grains of sand. They've never been trained. They have no exposure to their parents or siblings. They can do this out of the egg or even inside the egg. So the behavior is largely innate. Um, they only have a limited repertoire of skin patterns that they can adopt, as we'll get into. So it's not like they can change the skin pattern arbitrarily. They have a repertoire. So it's a limited set of patterns they can adopt. So the fact that it's innate and limited means that the solution might be simple. So akin to them playing some sort of visual trick to our on our visual system, sort of like a natural version of an illusion. Evolutionarily, these animals, the cephalopods, diverged from our own vertebrate lineage about 550 million years ago. So this means our common ancestor was a small marine worm that didn't have much in terms of a brain or complex behaviors. Uh, Therefore, the complex brains and behaviors of cephalopods and of vertebrates like ourselves are largely independently evolved. And any commonalities that we can find between these groups represents convergent evolutionary solutions to general problems facing animals. So, by studying camouflage and the different aspects of it, which have many parallels with complex behavior in vertebrates, so vision and motor control, decision making, etc., we can try to understand general properties of brains and behavior that would be difficult to study by looking at one evolutionary lineage in isolation. So currently, I think of camouflage as somewhat analogous to a variational autoencoder, which is a machine learning model. So here's the toy model currently is the animal take images from their environment using their eyes and visual system. And from these images, they're extracting some set of unknown summary statistics. These are the visual textures, and that these map somewhere deep in the animal's brain, which have a latent representation, some abstract representation of skin patterns. And these skin patterns are then rendered using the animal's motor system onto the ultimately the animal's skin, producing the camouflage pattern we can see in the animal's behavior. So if you're familiar with a generative adversarial network, it's another machine learning model. I think this is the even more precise analogy. We can think of the predators of cephalopods and the cephalopods themselves as playing an evolutionary game or an arms race, where the cephalopods are trying to generate realistic skin patterns, and the predators are trying to discriminate uh, between background textures, visual textures, and the disguised cephalopods. So over evolutionary time, we could train the cephalopod to produce more and more realistic skin patterns, and the predators to perceive and discriminate visual textures better and better. And I think what we see now in extant predators and in living cephalopods is the result of such an evolutionary arms race. So, okay, so that's how I think it works at a high level. Let's now zoom in and get into the details. So if we zoom in on a camouflaging cephalopod, this is a cuttlefish, we can see that the skin pattern is composed of tens to sometimes hundreds of thousands of specialized pigment cells called chromatophores. And these can occur in a couple different colors, depending on the species, and they can be expanded and contracted under control of these animals' brains. So, how it works is that every chromatophore is surrounded radially by a series of tiny muscles. So here's a single chromatophore. Remember, here's the scale. So it's like 25 micrometers across this chromatophore. And you see the muscles surrounding each chromatophore um, stained with actin. So you can see it here. Now, the motor neurons project all the way from the animal's brains to the muscles surrounding small sets of chromatophores and synapse onto them. So each motor neuron connects to a local group of chromatophores, and each chromatophore can receive input between 1 and 3 motor neurons in cuttlefish and octopus. Uh, and so when the motor neuron fires action potentials, it contracts the muscles surrounding each chromatophore and expands the chromatophore. And when the action potentials stop, the chromatophore contracts back to reveal the white skin underneath the set of chromatophores. So because mechanistically, this is how camouflage is generated in these animals, we reasoned that if we could track the expansion state or the area of the chromatophores, we not only have a quantitative readout of this amazing behavior, but also we'd be making a indirect measurement from the large population of motor neurons that are causing the variations in chromatophore area that we see over time. So to do this, we constructed an array of cameras. So this is 25 4K cameras uh, uh, put into a square array. And we mounted this array of cameras on a rail system overlooking an animal's tank. So this allows us to get the resolution needed to see the individual chromatophores, but have a large field of view so we can capture the entire animal as it swims around and camouflages to the background. And to deliver different visual texture backgrounds, we printed different images, visual textures, onto a fabric roll that allowed us to roll the different stimuli into the tank that the animal could camouflage to. So this allowed us to get two sorts of images. One is the image of the camouflaging animal in full, and the second one is zooming in and seeing a large population of chromatophores that compose the camouflage. So we did different sort of analyses for these two different kinds of images that we captured at the same time. So now I'll tell you first about the whole animal camouflage and a little later about our single chromatophore measurements. So we set out to first ask, what is the space of cephalopod camouflage? That is, what is the totality of the patterns that the animal can adopt or can be approximated at least. So to do this, we delivered a range of visual textures. So these are 30 different textures, artificial and natural, chosen to sample a range of different spatial scales. And we remember we printed these on fabric and rolled them into the tank to present to the animal. Now, previous work had suggested that the space of patterns is actually quite simple, that the animals can adopt three different patterns with some variation. A uniform, mostly white pattern, modeled pattern that looks more like sand, and a disruptive pattern that looks like with high contrast items like stones. So we thought that this is likely an oversimplification of the system. And we were interested in seeing whether we could bring a more quantitative approach to the problem and reveal more of the structure that we thought would be present. So to do this, we took our images of the animal, camouflaging to our printed visual textures, and first did some image processing to identify the animal and using a neural network, and then rotated the images and standardized them to produce a set of a large set of images of the animal always facing north and doing different camouflage patterns. So how do we describe the camouflage pattern? Besides, we can describe it in terms of the image, but how do we summarize it? So what we settled on was using a neural network. Previous work had shown that the VGG19, which is a deep convolutional neural network pre-trained on lots and lots of images from the ImageNet database, the deep layers of this network had previously been shown to provide a useful parameterization of visual textures in the world. And we reasoned that we have here the animals adopting visual textures in order to blend in with the camouflage to visual textures. So we could use the VGG network to parameterize the camouflage patterns from cuttlefish. So we take these images, pass them through the network, and take the fifth convolutional layer as our description of camouflage. So we go from 4,000 by 3,000 pixels in the image to 512 dimensions for describing this camouflage. So using this description, we could take lots and lots of images of many animals camouflaging to our 30 different visual textures, and pass them all through our neural network and look at the space of images that comes out. And we found that the space was about 50 dimensional. So this is much more complicated or much more high dimensional than had previously been described, but much lower dimensional than it could be in principle. Remember, these animals have hundreds of thousands of chromatophores. So in principle, it could be a hundreds of thousands dimensional space. It's not, it's 50 dimensions, at least as best we can estimate it. We can image this high dimensional space using UMAP, so it's a nonlinear dimensionality reduction technique. So here is the space of camouflage patterns in cuttlefish as we measured it. Each image here, each dot here is an image, and different regions of this space correspond to the animal adopting different skin patterns. So we can see that here by pulling out images from different regions of this space. So here's from one, here's 9 images from this region 1, here's 9 images from region 2, etc. We can see here that all the camouflage patterns within a block here resemble each other, but they differ from the other blocks. So this says that our description is doing a good job of teasing out these different camouflage patterns. If we look at the difference between, for example, block 2 and block 3, you can see by eye that there is a difference, but I would be hard pressed to describe in words what that difference is. There's some different pattern elements are darker here than here, perhaps, but the network can tell these things apart. So this highlights the importance or the benefit of taking this more quantitative approach. Okay, so when we deliver different visual textures to different animals, we could get somewhat reliable responses. So These are three different cuttlefish on the y-axis here, camouflaging to different visual textures and adopting similar patterns across. So this means that there's a conserved repertoire of skin patterns across animals, and they can change them to match the different textures pretty well. So we can quantify that here. So we can look at the top principal component, but this works for the top few principal components of the visual textures of the background, comparing that to the visual texture that the animal adopts in that background. And the three different colors are three different animals. And we can see here a strong correlation between the background visual texture and the skin texture of the animal after these spaces have been aligned. So this tells us that the animals can precisely match or tailor their skin pattern to different visual textures. So this 50-dimensional space that the animals have with camouflage patterns, they seem to be able to move around this space and stop at different arbitrary points in that space. It doesn't seem to be discrete. We can see that better perhaps here, where we reduced the background to just checkerboards of different spatial scales. So from very fine checkerboards to very coarse checkerboards. And again, as we saw, we can study how the animal changes its camouflage pattern in response to these different spatial frequencies. We can see for three different cuttlefish now, the animals will smoothly transition from one camouflage pattern to another as we vary the spatial frequency of the checkerboard. There's not a discrete jump from one camouflage pattern to another, as you would expect if the animal had a discrete set of patterns. So it seems that there's this high dimensional pattern continuum, and the animal can smoothly move around it. Okay, so now moving in to look at the individual chromatophores, taking advantage of our high resolution videos. We can use computer vision and neural networks to align these images and segment out the chromatophores and just isolate the fluctuations of individual chromatophores, removing larger scale movements like the animal swimming or breathing. And this allowed us to study chromatophore dynamics. So we've done many different things with this sort of data of tracking chromatophores over time. This is published if you're interested in checking it out further. We can do things like study in the context of camouflage as the animal moves from one camouflage pattern to another in this pattern space. What are the individual chromatophores or groups of chromatophores doing during these camouflage pattern transitions? We can also study, by tracking chromatophores over weeks and months, the developmental process, so its behavior at a very slow scale, as the animal continuously grows and new chromatophores are insert themselves into the skin, forming this really elaborate and interesting pattern over the animal's life. Another thing we can do is look at the correlations of the fluctuations, so the chromatophores expanding and contracting together, and use these sort of measurements to infer elements of a hierarchical motor control strategy. So let me tell you a little bit more about this piece here. So if you look at the chromatophores in detail and track them, so here's a small patch of skin. So here's the scale bar is 50 micrometers. We have a group of chromatophores here in red that show really strong correlations. So here are the time courses of the chromatophore fluctuations. So each trace here is a chromatophore, and this the scale bar is 5 seconds. So this is over 30 seconds or so. And the peaks here are the chromatophore expanding and then contracting. We see that all the red chromatophores here show correlated expansions. They all fluctuate together. Now there's blue chromatophores that are right next to it. There's the nearest neighbor chromatophores that sometimes show correlations, but sometimes don't. They're doing something else. So we think that there are these groups of chromatophores that show strong correlations because they're receiving input from a single motor neuron. Every time the motor neuron spikes, all the chromatophores that motor neuron synapses onto, or the muscles the surrounding the chromatophores the motor neuron synapses onto, expand or contract, and the chromatophore expands. And this is why we see the co-fluctuations of small groups of chromatophores. So if we look at the animal's back here, but now it's single chromatophore resolution. So here I'm just zooming in on the center of the back. We can identify large numbers of these putative motor units. So these are groups of chromatophores that we think are receiving common drive from a motor neuron. And they form these little spatially localized clusters in these different colors or different of these putative motor units. So we think this is the looking one level down from the chromatophores to inferring motor neuron activity. Now, if we cluster these clusters, we can do this hierarchically, continuing to cluster less and less correlated chromatophores. And if we look at the clustering a little higher up in this dendrogram, we can find groups of chromatophores that are more spatially extended but are still co-fluctuate, but at less frequently than at the level of the individual putative motor neuron units. So these we think are reflecting a premotor neuron that is synapsing onto a group of motor neurons that is synapsing onto this group of chromatophores that we are measuring. And we can play this game looking at higher and higher levels of the hierarchical clustering until we get towards the top, where we see large groups of chromatophores that show a heightened level of correlation and form the macroscopic patterns that we can see by eye on the animal's back, like this white square. This is all showing one big cluster. So what we think we're doing is we think we're looking deeper and deeper into this animal's motor control system, which forms a hierarchy from abstract, large pattern elements towards the more and more refined motor neurons and ultimately synapsing onto the small groups of chromatophores that compose the pattern. So we think we're looking back from the skin back into the brain through this image rendering step towards what we think of as somewhere deep in the brain that's able to spontaneously generate skin patterns. And we think that such a region exists because of our work on sleep, which is unexpected connection. So let me tell you about that for a little bit. So when you went to sleep last night, your brain alternated between two stages, REM sleep, where you have rapid eye movements and the most vivid dreams, and slow wave sleep, where your brain the cortex undergoes these large, slow oscillations, synchronizing much of the brain. So why we have two stages of sleep and why we alternate between them throughout the night, I think is one of the great mysteries of science, definitely neuroscience. And it's not known. There's many ideas. But evolutionarily, the evidence points to it being very important. And this is because two stages of sleep that resembles our own two stages, REM sleep and slow wave sleep, is found across many different vertebrate groups. So the parsimonious explanation is that it evolved some 400 million years ago, at least, and has been maintained over these hundreds of millions of years and in such diverse animals because it's doing something very important. We don't understand what it is. It's in this context that it's interesting to look at the cephalopods because, as I said, they diverged from our own lineage so long ago, and they've independently evolved large brains and behaviors, that if we could find anything that resembles the two stages of sleep in a cephalopod, it would suggest that two stages of sleep can convergently evolve multiple times, and is therefore something that's quite fundamental, similar to how a vision evolved at least 5 times, or camera type eyes evolved at least 5 times in evolution, because it fills such an important functional role. So being in Okinawa, we're able to go out at night in the winter and gather small octopuses in the tidal pools around the institute. And we wanted to study sleep in octopus. So these are Octopus laqueus. It's a small species of local octopuses. And they're nocturnal, so they rest all day in the laboratory. And this is a top-down view of a resting octopus. This is looking at the mantle only. So the eyes are up here, and the arms are tucked up around the mantle. And they look like this all day long as they rest, and they're active at night. But we found when we studied them that periodically they would do this dramatic color changes. So this is over about a minute. The animal rapidly transitions between different skin patterns in a behavior that we term active rest. And this the speed of these transitions is nothing like we see when the animals are awake. It's very rapid. And it's accompanied by different movements that I'll tell you about. And after this about a minute of active rest, the animal goes back to its normal quiet rest, where it's calm and all white because the brain-controlled skin patterns are not active. They're not turned on during quiet rest. So we could quantify this behavior simply by just looking at the average intensity of each image. So we can see the animal starts off all white, and then it goes through this rapid skin pattern changes during active rest, and then settles back down to another bout of quiet rest. We found that this behavior repeats. So throughout the day, you have a bout of active rest that's separated. It's followed by about an hour of quiet rest before another bout of active rest. And this goes on and on all day long before the animal wakes up at night to hunt. So besides the rapid skin pattern changes, active rest we found was accompanied by rapid eye movements. So this is quantifying active rest compared to quiet rest. So there's more eye movements, there's more body twitches, the breathing rate of the octopuses goes up, and also the variability in breathing. And if you know the sleep literature, these are all behavioral hallmarks of REM sleep, or this more active wake-like sleep in vertebrates. So is active rest in octopus a stage of sleep, a second stage of sleep? So to test this, we relied on the evolutionarily conserved behavioral criteria for sleep. So it turns out that there are three behavioral criteria that characterize sleep across the animal kingdom, from jellyfish to humans. The first of these is whether the behavior is rapidly reversible. So this separates sleep from things like coma or death. Sleep is rapidly reversible. So to test this, we took the animals during rest, so either quiet or active rest, and we put an electromagnet on the side of the animal's tank. And this allowed us to deliver a controlled tap, a mechanical tap to the side of the tank. And so we could track the animal's movements with optic flow. So here's the optic flow when the animal's resting, and then we give the tap here, and it does a startle jump and changes its color. So we can record this movement. This behavior is rapidly reversible. So we found that when the animal is in active sleep in red, quiet sleep in pink, or wake in this sort of light pink, when we really tap the tank really hard or strongly, the animal would always react. So this both active and quiet rest is rapidly reversible. We then dialed down the strength of our stimulus about 80-fold, and we could find a regime where the animals will react when they're awake, but they didn't react when they're either in active rest or quiet rest. So this means that both these behaviors, active and quiet rest, come with a heightened arousal threshold. This is the second behavioral criteria for sleep. The third criteria is whether the behavior is under homeostatic control. So that is, if we prevent the animals from entering this putative sleep stage for a period of time, and then let them freely behave, if it's sleep, we should expect that they would do more of this sleep stage. So just like if you pull an all-nighter, you get really, really sleepy. There's a drive to sleep that is only satisfied by actually sleeping. So remember, these animals are resting all day and they're active at night. We can track them under red light, which they're not very sensitive to. So this is tracking the number of active rest bouts over 36 hours. So this is a nighttime, daytime, nighttime experiment over many octopuses. And we can see that the octopuses do many more of these active rest bouts during the day, not very many at night when they're active, they're running around hunting for crabs. So we then, my student, graduate student Aditi, continuously bothered these animals with a paintbrush for a couple days, and then let them freely behave. And we saw what they would do. We found that that night, the animals did many more of these active rest bouts than normal. And the following day, they did just as many active rest bouts. But then 2 nights later, following the deprivation, they again did more active rest bouts. So active rest is under homeostatic control. It comes with a heightened arousal threshold, and it's rapidly reversible. So it meets the 3 behavioral criteria. And so this is a second stage of sleep. Octopuses have 2 stages of sleep, like us, and an active wake-like stage and this quiet rest-like stage. So their active sleep resembles REM sleep in many ways. We did brain recordings to show that there's wake-like neural activity accompanying active sleep. There's all these behavioral criteria or behavioral correlates that looks like REM sleep. But one big difference is that these animals have their brains directly connected to their skin, and they're doing these skin patterns. This is something that we don't see in vertebrates, of course. And I think this is the most interesting aspect of active sleep in cephalopods. So we can find the animals do waking patterns. We can find the very similar patterns show up during active sleep. So here we have 2 images: waking image in purple, active sleep image in green. And here, after we've aligned these 2 images, this is the overlap image. So the white is overlap. We can see that the exact splotch pattern of expanded chromatophores matches between wake and active sleep. And we can do this for many patterns. So it seems that during active sleep, these animals are rapidly transitioning between around their waking skin pattern repertoire. So they're traversing this pattern space that we revealed in the first part of the talk while they're asleep. Why are they doing this? We're not clear. Of course, we're very interested in understanding, what's the reason why they do this, why they have active sleep, and why they do this skin patterning. But the fact that it exists suggests that there is an area in the animal's brain that's able to spontaneously generate skin patterns independent of visual input. And we're seeing the result of that after it's being rendered on the animal's skin. So this is how our work on sleep has influenced our view of camouflage. We think of camouflage as when the animal sees the visual environment, and this visual texture representation is biasing this generative mechanism that we revealed during sleep for the animal to produce the correct skin pattern in that visual environment. So that's our current view of how this works. And what we're interested in doing now is looking deeper into the brain, actually by doing neural recordings and trying to unpack this toy model and see really how the brain implements the different aspects of camouflage. So the story so far, and what I've told you about, is how camouflage in cephalopods forms this high-dimensional pattern continuum, and they're able to smoothly move around this space to precisely match with background images. It's a more complex system than had previously been described, which raises all sorts of interesting questions of how they do this without any sort of teacher, without any examples. How do they know? So, which features of this behavior are innate, and which are learned, and how is it learned? I think are really interesting questions to explore. Um, on the sleep side, we've described this active sleep stage in octopus. Cuttlefish do it as well. It has many parallels to vertebrate REM sleep, suggesting that this awake-like sleep stage evolved at least twice in evolution and likely goes hand in hand with having a large brain and complex behavior to perform some fundamental functions that I think are still mysterious, although there are many interesting ideas out there. Um, Regardless of the function, it's the sleep work suggests that the generative brain region must exist in animals, somewhere in the animal's brain. And we're interested in exploring where exactly this is and what format the latent patterns take in the animal's brain. And what I've hoped to have done is to convince you that these brain-controlled skin patterns in cephalopods provide really a uniquely rich read and direct readout visual perception in these animals. Um, and at a high level, I don't know if you notice the octopus in these images. Um, uh, the fact that cephalopods have independently evolved these their brains and complex brains and behavior uh, make them a really unique animal to study not just camouflage but any sort of complex cognitive ability because it allows one to compare and contrast with other complex organisms like ourselves or other vertebrates, and really delineate which aspects are general and shared across both groups, and which are specific to something about the vertebrate evolutionary tree or the cephalopod. With that, I'd like to thank everyone in my lab, our collaborators and funding sources, and thank you for your attention. Thank you.
That was such an interesting talk, and it's really interesting to see how these cephalopods can generate all these different patterns, and also like I didn't expect about them sleeping. We already have quite a few questions in the YouTube chat. So the first question is from Ethology Dave. Even though the camouflaging ability is innate, can cephalopod become better at it over time, or the opposite, the camouflaging mechanism deteriorate and can they become worse with time?
Yeah, it's a great question. I think likely the ability of the animal to choose which of its patterns that I think that the pattern repertoire is innate, but the choice of which pattern to adopt in which situation probably has a learned component. But we haven't really unpacked what aspects are learned versus innate. And we run into the problem of assessing camouflage quality. So when you see these pictures of the animal in the wild, it's the camouflage is quite excellent, and there's no doubt they're camouflaging. But in the lab, sometimes they do something that looks kind of camouflage-like to my eye, but not it's not as precise. In those situations, I don't know whether that's the animal trying to camouflage and doing a bad job because it's difficult or it hasn't learned it well, or it's just not trying. So this complicates the question.
Next question is from Valentine. How much do the results regarding patterns exhibited by the animals depend on the diversity of tested background? That is, how to characterize the range of all possible patterns to be tested. A bit analogous to making sure the whole parameter space has been tested.
Yeah, I think it's a great point. Likely, we are under sampling the range of visual environments that the animals can camouflage to, and Therefore, the 50-dimensional space that we uncovered is probably a lower bound on the true space. Maybe the sleep result, which also turned out to be around 50-dimensional, if we can assume that they're really exploring their full repertoire during sleep, we could use that as a more unbiased estimate of the repertoire. But I think the only way really to do it is have a really systematic mapping of their patterns throughout their natural environments. So this is field work, which we're interested in doing, but it's hard to get to find them and to really map fully. So it's a difficult problem, but I think it's a problem shared by, I think, every lab-based animal behavior study.
Then the next question is from David. Thank you for a fascinating talk. Many of the camouflage patterns seem to be symmetrical vertically. Is there a physiological reason for this?
Um, the eyes are lateral and project ipsilaterally, so just to the same side of the brain, and the brain is symmetric left-right and projects left-right, so that they can actually adopt two different patterns along the midline. They can't split their patterns in other ways. So I think that this is why you have this this impression of symmetry. They can break it just this one way. Most of the time they don't. It stays symmetrical left-right.
Uh, the next question is from Catherine. Hi, this was an inspiring talk. Thank you. Is there a way you could dye a motor neuron to see the chromatophores it is connected to validate your hierarchical model?
Um, Yeah, so I think it'd be hard to do it with dye. What we did was stimulate electrically single chromatophore or single motor neurons and showed the size of the motor unit, so the collection of chromatophores that receives input from that single motor neuron to validate in separate experiments in this isolated skin that our estimate of motor units in vivo is a ballpark around the correct thing. But to do it over all chromatophores in the intact animal, I think is really difficult.
Uh, Next question is from Valentine. Regarding sleep, you mentioned interesting ideas to explain the potential functions of these cycles. What's your favorite? Is there indeed coevolution of this feature?
I don't know whether there's coevolution. That's something we're interested in exploring. My favorite idea currently for why we have this active or wake-like sleep stage, and maybe why octopuses have it as well, is that this provides a mental simulation of a waking experience that's risk-free. So we can simulate things that you're not really sure yet how to do without risking getting eaten by predators. So this ability to roll out simulations and train up the system offline, I think, is something that we've seen in recent AI efforts, exploring as a way of boosting the efficiency of training data. And I find it a really compelling idea.
Next question is from Lachlan Walsh. Great talk. The PC score of multiple cuttlefish smoothly transition between patterns, but there are some strong individual variation. Could this be from variation in visual perception or differences in chromatophore control capabilities?
Um, I think it's hard. I mean, another possibility is that it's just individual, individual difference in what they want to do. There's some individuality in their behavior. So to trace the origin of that individuality, whether it's on the visual perceptual side, the motor side, or somewhere in between, so their decision making is an interesting question, but I don't think we can tell just from the behavior alone. We'd have to do, I think, some follow-up experiments.
Um, Next question is from Carlos Gueller. Hi Sam, beautiful talk. You mentioned some hierarchical organization in the chromatophore activity. If you record it from the optic lobe while the animal is camouflaging, would you expect to see this hierarchy of activity already at this level? Or is it a consequence after the dimensional reduction of visual input?
Um, let's see. So the question of whether the representation of skin patterns, this latent representation that we think exists, is present in the optic lobe, which is it's a visual area or a visual motor area. It's an empirical one. I don't know. I think likely the representation goes from being more visual to being more motor, but I'm not sure whether there's going to be a sharp dividing line saying this is visual and this is motor, as you would in construct in an engineering sense. Uh, the data from other animals says that things get more mixed up. So maybe we start seeing um, uh, the precursors of something more motor-like in the context of visual neurons in the optic lobe. Is one possibility, or maybe the motor stuff starts shading in a little bit downstream. We're not sure. Um, is the uh, uh, is the result is it an artifact of the dimensionality reduction? I think uh, the analysis was a hierarchical clustering of the correlation patterns of the chromatophores, and this, by definition, will have a hierarchical answer. So I don't think we can definitively say that the motor control is hierarchical due to the analysis I presented. But there's other pieces of evidence, like stimulation results from more deeper in the brain seems to produce full patterns, when stimulation closer to the periphery produces these more localized motor neuron-like patterns. So things like that suggest hierarchy. So maybe that hopefully that addresses some of the question.
Uh, Another side question from Carlos. Can their mantle show things like emotions other than visual imagery?
Um, I think so. I think you get into kind of a tricky business to what exactly do you mean by emotion. But yeah, there's definitely more coming out onto the skin than just camouflage. They do patterns in situations where it doesn't make sense for camouflage. Whether this reflects their emotion is a fascinating question. But I would be at a loss to know what's the knockdown experiment to show its emotion and not some other internal state. Or if emotion just means internal state, then yes, it's emotion.
Another question from Catherine. The similar patterns during active sleep compared to the wake state made me think of memory consolidation, in which waking experience is reactivated in the sleeping brain. Do you think there could be a similar mechanism at play?
Yeah, so this was our first guess that it's something analogous to hippocampal replay, where the waking experience of the animal is then reactivated preferentially that night during well, in the rat case, it would be during slow wave sleep, but in this case, during active sleep. Um, we've done some experiments that suggest that this is not the case. So we've done when we bother the animal, they produce a characteristic pattern. Maybe it's emotion. They do this sort of polka dotty pattern. And so when we bother them all day and they do this pattern a lot, then we look the following night whether there's more of this pattern or it's we pass through it more often. We don't seem to see a difference. So uh, it, the sleeping patterns the active sleep patterns, may represent waking experience at a longer time scale than we've explored, or it could represent some sort of developmental process. We're not really sure what it represents currently. And this is active research.
Um, and I think we are also nearing our end time. And thank you again, Dr. Rader, for giving us such a great talk and answering all the questions. And I'm sure if people have more questions, they can contact you through email and you'll be happy to answer them. And again, thanks to all of you for joining us for this exciting talk. And feel free to keep looking at our socials and looking for further seminars. And we are also going to have our conference coming up soon. So make sure to submit your abstracts for that too. Thank you again and hope to see you soon. Thank you.
Thank you very much.